Looking at our third biological molecule, the proteins. Proteins are composed of monomers called amino acids. It's an extremely important macromolecule. More than 50% of the dry mass of a cell is protein. Dry mass is referring to the cell once all of the liquid has been removed. There are many functions of proteins. All enzymes are proteins: peptide, DNA polymerase. Proteins are essential to cell membranes. Many hormones are proteins, such as insulin and glucagon. Oxygen-carrying pigments, hemoglobin and myoglobin. Antibodies, which acts as a defense for our wellness. Structural component, such as bones, collagen, keratin. Muscle contraction by actin and myosin. Storage, such as casein in milk and albumin in egg whites. Proteins are carriers and transporters, such as with hemoglobin and aquaporin. And there's cell communication, working with signals and receptors. All amino acids have the same general structure. There's the central carbon atom, and then an amine group attached. The amine group is your NH2. And then you have a carboxylic acid group attached. That's your carbon with that double bonded oxygen and a single bond oxygen with the hydrogen, the hydroxyl group here, and double bond oxygen here. The R can be exchanged for any amino acid. In this example at the bottom, you have the same structure again. You have your amino group, your carboxylic acid, all bonded to that central carbon atom, and then the R, which can change depending on the amino acid. This example has glycine as the amino acid that's in place of the R. Again, that can change out. There are 20 different amino acids, all with a different R group, commonly abbreviated with 3 letters, such as glycine is represented with GLY, or sometimes they'll do a single letter, such as just a capital G. We saw the 3-letter abbreviations when we looked at protein synthesis in pre-ACE biology. This was that amino acid chart that you would use when you were looking at protein synthesis. The peptide bond. One amino acid loses a hydroxyl, that's that OH group, from its carboxylic acid, while another amino acid loses a hydrogen from the amine group. This leaves a carbon atom free to bond with a nitrogen atom, forming a link between the two called a peptide bond. Peptide bonds are strong covalent bonds. When a peptide bond is formed, you're going to have a condensation reaction, so water is going to be removed. 2 amino acids combining together, we get a dipeptide. Di, remember, means 2. More than 2 amino acids, we will have a polypeptide. A complete protein may contain just one polypeptide chain or many that interact with each other. In living cells, ribosomes are the sites where amino acids are joined together to form peptides. This reaction is controlled by different enzymes. Polypeptides can be broken down by hydrolysis to amino acids. This happens naturally in the stomach and small intestines during digestion. The amino acid released is absorbed into the blood. This is showing you 2 amino acids bonding together. Here's one amino acid and the second amino acid. The hydroxide of one amino acid and the hydrogen on a second amino acid are going to bond together to form water. It's releasing water. Remember, that's known as a condensation reaction. And then when the two amino acids are bonded, that's going to form the peptide bond. Here's your carbon with your double bond oxygen. Notice that's what you have here. And then on the second amino acid, you have the nitrogen with the hydrogen, which you have right here. And then that carbon atom and the nitrogen of the second amino acid, they bond together, forming a peptide bond. This is known as a dipeptide because there's two amino acids. Di means two. And then notice you have that NCC, NCC. You have that on here as well for the amino acids. Nitrogen, carbon, carbon is typically the atoms that you see for identifying amino acids and then proteins. The reverse is true where water can come in as hydrolysis. Water would come in, breaks this bond, this peptide bond, allowing the hydroxyl group to bond to one amino acid and hydrogen to the other. Taking a look at the structures, polypeptide chains may contain several hundred amino acids linked by peptide bonds. So in between each amino acid, you would find a peptide bond. The particular amino acids and their order in the sequence is called a primary structure of the protein. The sequence determines the structure, and the structure determines the function. Change the sequence, and that changes the structure, which changes the function. There are enormous numbers of different primary structures possible. A change in a single amino acid in a polypeptide can completely alter the structure and function of the final protein. The secondary structures. The particular amino acid in the chain have an effect on each other even if they are not directly next to one another. This is because the polypeptide chain can bend back on itself. Polypeptides often coil into a corkscrew shape called an alpha helix. This secondary structure is due to the hydrogen bonding between the oxygen and the double bond carbon-oxygen group of one of the amino acids and the hydrogen of the NH group on the amino acid 4 places ahead of it. And by that you can see where they have the amino acids numbered 1, 2, 3, and 4. And so you have these hydrogen bonds forming from the first one to the fourth one. They are easily broken by high temperatures and pH changes. Hydrogen bonding is also responsible for the formation of a beta pleated sheets. These are looser and straighter than an alpha helix. Again, they can easily be broken by high temperatures and pH changes. You'll look at the diagram, it kind of looks like a pleated sheet. Some proteins show no regular arrangement. It depends on which specific R groups are present and the attraction between the amino acids in the chain. In diagrams, the beta pleated sheets are represented by arrows and random coils as ribbons. The alpha helixes are represented by coils or cylinders. In many proteins, the secondary structure itself is coiled or folded. Shapes may look random, but they are very organized and precise. The way in which a protein coils up to form a precise 3D shape is known as its tertiary structure. There are 4 bonds to hold tertiary structures in place, and you guys need to know all 4 of these. The first one is a hydrogen bond. It forms between the R groups. The hydrogen bonds are weak in isolation, but many together can form a strong structure. Again, that dotted line is representing the hydrogen bond. The disulfide bonds form between two cysteine molecules. They contain sulfur atoms. So again, you have the sulfur bond, that disulfide bond here. The last two ionic bonds form between the R groups containing the amino and carboxylic groups. They can be broken by pH changes. And then the fourth one is the weak hydrophobic interactions. They occur between the R groups, which are nonpolar, therefore they're hydrophobic. The two figures show the complex way in which secondary structures coils or folds to form a tertiary structure of a protein. Now looking at the quaternary structures, most protein molecules are made of 2 or more polypeptide chains. Example, hemoglobin, which has 4 polypeptide chains. The association of different polypeptide chains is called the quaternary structure. Chains are held together by the same 4 types of bond as the tertiary structure. Quaternary does not mean there are 4 polypeptide chains. A protein whose molecule curls up into a ball shape is known as a globular protein. Globular proteins usually play a role in metabolic reactions. Their precise structure is key to their function. Enzymes are globular proteins. Globular proteins usually curl up so that their nonpolar hydrophobic R groups point into the center of the molecule, away from the aqueous environment. Globular proteins are usually water-soluble because their water molecules cluster around their outward-pointing hydrophilic R groups. And you can see in this diagram where you have the hydrophilic R groups represented in this turquoise, and then the hydrophobic R groups represented inside with the kind of yellowish color. Hemoglobin is the oxygen-carrying pigment found in red blood cells and is a globular protein. It's made of 4 polypeptide chains and has the quaternary structure. Each chain is known as globulin, and there are many types of globulins. 2 types of globulin used to make hemoglobin. Hemoglobin is going to contain 2 alpha globulins. They make the alpha chains, and 2 beta globulins, which make the beta chains. Hemoglobin is nearly a spherical shape due to its tight compaction of the polypeptide chains. The hydrophobic R groups point towards the inside of the protein. The hydrophilic R groups point outwards. Hydrophobic interactions are essential in holding the shape of the hemoglobin. The outward pointing hydrophilic R groups on the surface of the molecule are important in maintaining its stability. Sickle cell anemia is a genetic condition in which one amino acid on the surface of the beta chain, the glutamic acid, which is a polar molecule, is replaced with a valine, which is nonpolar. Having that nonpolar or hydrophobic R group on the outside of the hemoglobin makes it less soluble, and this causes the blood cells to have the wrong shape. Each polypeptide chain of hemoglobin contains a heme group. It's an important permanent part of a protein molecule, but it is not made of amino acids. It is called a prosthetic group. Each heme group contains an iron atom that combined with one oxygen molecule. A complete hemoglobin molecule can therefore carry 4 oxygen molecules, which is 8 oxygen atoms total. It is the heme group which is responsible for the color of hemoglobin. The color changes depending on whether or not the iron atoms are combined with oxygen. If they are, the molecule is known as an oxyhemoglobin and is bright red. If they're not, the color is a darker, more bluish red. Proteins that form long strands are called fibrous proteins. These are usually insoluble in water. Most fibrous proteins have structural components in the cells, such as in keratin and collagen. Collagen is the most common protein found in animals. It is approximately 25% of the total protein in mammals. Insoluble fibrous protein found in skin, tendons, cartilage, bones, teeth, and the walls of blood vessels. It is an important structural protein. Collagen consists of 3 helical polypeptide chains, each a helix, that form a 3-stranded rope or triple helix. 3 strands are held together by hydrogen bonds and some covalent bonds. Almost every third amino acid is glycine, which is a very small amino acid. This allows the strands to lie close and form a tight coil. Any other amino acid would be too large. Each complete collagen molecule interacts with other collagen molecules running parallel to it. Covalent bonds form between the R groups of the amino acids lying next to each other. These cross links hold many collagen molecules together side by side, forming fibrils. The end of a parallel molecules are staggered to make the fibrils stronger. Many fibrils together is going to make a fiber. Many collagen molecules make up one collagen fiber. Looking at the structure of collagen, here you have one of the helixes that has been stretched out. You'll notice that every third amino acid is glycine. 3 of those helixes are going to combine together. They are bonded with some covalent bonds and hydrogen bonds. And then several of these 3 helixes is what we're looking at here. Each one of these is representing 3 of the helixes bonded together. They're linked side by side. You have some cross links between it, giving it more strength. And then as these are bonded together, many of them are going to combine together to make fibrils. And then the fibrils are going to combine together to make the fiber. This is giving you that representation where you have one of the chains and then 3 of them, how they get tight, just giving you a different image. There's a tremendous tensile strength within those fibers. They can withstand large pulling forces without stretching and breaking. They're also very flexible. The Achilles tendon, which is almost pure collagen, can withstand a pulling force that is about 1/4 of that of mild steel. The collagen fibers line up in the direction in which they must resist tension, such as parallel bundles along the length of the Achilles tendon, cross-layered in the skin to resist multiple directions of force. Okay, you need to know again how to test for the biological molecules. So for testing for proteins, you would use the Biuret solution. When the biuret solution is positive in the protein, it's going to turn a purple color. All right, 3 questions that I would like you to answer within your notes. For this second one, you are going to need the table that has been included in your guided notes.